First, water is a single molecule. Then it splits into hydrogen, protons, electrons, and oxygen. Oxygen is given off as a byproduct, and the electrons are then used in photosystem 2. Photosystem 2 is where chlorophyll pigments absorb sunlight to excite the electrons. After this, electrons go through an electron transport chain with cytochromes that help pump hydrogen plus ions into the thylakoid space. This creates a concentration gradient in the thylakoid space. This will cause the hydrogen plus ions to diffuse through ATP synthase. This in turn drives ATP synthase to produce ATP through photophosphorylation. After going through the electron transport chain with cytochromes, the electrons need to be re-excited and as a result go through photosystem 1. To create more ATP in cyclic electron flow, the electrons will go back through the electron transport chain to once again make ATP for the Calvin cycle. If the electrons do not go through cyclic electron flow, they go through non-cyclic electron flow. The electrons would instead go through another electron transport chain without cytochromes after being excited by photosystem 1, and finally pairs up with NADP plus and hydrogen plus ions to make NADPH. This, along with the ATP, is finally ready to be used in the Calvin cycle. To begin phase 1, the carbon fixation of the Calvin cycle, there are 3 molecules of 5-carbon sugar, RUBP. This sugar is already present in the Calvin cycle and does not need to be added. Then 3 molecules of carbon dioxide enter. This CO2 enters through the stoma on the underside of the leaf. The enzyme Rubisco puts the CO2 molecules into the RUBP molecules, making them unstable 6-carbon sugars, which now want to split. They break into 6 3-carbon sugars called PGA. In phase 2 of reduction, all 6 molecules of PGA are phosphorylated by ATP and reduced by NADPH. Basically, ATP gives each PGA a phosphate group and NADPH gives each PGA an electron, which is followed by a hydrogen ion. This turns the PGA molecules into G3P molecules. This also regenerates ADP and NADP+ to be used again in the light cycle. One of the G3P molecules goes into becoming half of glucose. Once the cycle happens another 3 times and another G3P molecule is made, an entire glucose molecule can be formed. To complete the cycle, we have phase 3, regeneration of RUBP. The remaining 5 G3P molecules are reorganized into 3 molecules of RUBP from the very first step. This regeneration requires ATP, which is made in cyclic flow of electrons in the light reaction. The 3 molecules of RUBP are ready to accept CO2 and complete the cycle all over again.